Started. Welcome to our first review for our final exam. I will have final exam information posted in the announcements sometime today to let you know exactly what's going to happen. I will also be going through your grades to see if there's stuff that didn't get credited that was turned in, extra credit, that kind of thing. I'll also be posting what you can do for extra credit if you haven't been able to do the other extra credit assignments. And so all that is forthcoming. Ha, get it? And I wore my Star Wars shirt. Yay. May the 4th be with you.
Can you remind me what the extra credit things were? I was trying to remember.
So we had the Tesla thing and blood drive and the help on the STEAM festival thing.
Okay.
Yes. So, okay. So, did anybody look at the little study guide thing?
Yes.
Was there anything in particular that you needed me to go over? And I know it's like bullets, you know, it's just, those are the topics. Then you need to know everything about them, for lack of a better way of explaining that. If you go through your objectives for each one of the lectures, that will help too. So I highly suggest that you do that. Okay, so anybody have anything to begin with? Otherwise, I'll just like hit some things as we go down through the list. I guess I could share this. I'll share it. You don't need to be looking at me. Okay. All right, so here we go. The very first item is, what are hormones? So we need to remember hormones are chemical messengers that are going to be secreted by endocrine glands directly into the bloodstream. And then remember that hormones are going to allow a response to be last longer and more widespread throughout the body because it is in the bloodstream. And then because hormones have half-lives, they need to be removed from the bloodstream in order to cease their function. And so there will be certain lengths of time that it'll take particular hormones to be removed from the blood in adequate amounts that they don't affect the receptors anymore, or their target tissues. Go back through your hormone chart and find out where they come from and what they're going to do. We should be able to rattle off all of those hormones now and be able to explain their functions. So review that. So the hormone chart. Okay, receptors, remember that all hormones require a receptor in order for them to do their job. And there are two kinds of receptors. There are receptors that are going to be on the cell surface, and there are intracellular receptors. The ones on the cell surface are generally going to elicit rapid responses and require a second messenger. So when they bind to the surface, then they're only going to activate processes that are going to happen within the cell by the use of G protein. And then a second messenger, like calcium is a second messenger, cyclic AMP is a second messenger. Yeah, that's good on those. And then the intracellular receptors are going to be specific for the steroid hormones and thyroxine. So those are the ones that are going to go on the inside. So aldosterone and cortisol, and then estrogen, progesterone are going to go into the cell and find the receptor either in the cytoplasm or in the nucleus or already bound to the DNA. Let's see. So we're going into that second messenger's gene activation. Generally, the responses elicited by gene activation are going to be longer because, you know, you got to make a gene product, but sometimes they can be fast too, and we'll get into what those are in just a minute. Tropic hormones. So remember, a tropic hormone is a hormone that is going to stimulate the production of another hormone by a gland. The tropic hormones will be all of the hormones from the hypothalamus that act on the anterior pituitary. So the releasing and inhibiting hormones, releasing hormones. So GnRH, that's the only releasing hormone that you know a name of, is gonadotropin releasing hormone that acts on the anterior pituitary, stimulates FSH and LH production. And let's see what other tropic hormones are there. So from the anterior pituitary, there are 4 tropic hormones, FSH and LH, that I already mentioned. ACTH and TSH are the tropic hormones. Stimuli. So there are 3 kinds of stimuli for hormone release. They are hormonal stimulus, obviously the tropic hormones. Humoral, that's the main one. So various levels of substances within the blood will elicit release of a hormone. Calcium levels, water levels, sodium levels. Let's see, what else? And then the neural stimuli, so the stimulus from sympathetic stimulation to the adrenal medulla, or the stimulus of uterine contractions going back to the hypothalamus and the posterior pituitary to release oxytocin during that positive feedback cycle of labor. And nursing, breastfeeding. So that sucking action on the breast is then going to go up to the anterior pituitary. I guess there's not. Is there a prolactin releasing hormone? Can't remember if there's one from the hypothalamus or not. Obviously, that's not a test question. But anyway, so it's going to go to the anterior pituitary to increase prolactin production so that you make more milk as your baby nurses more. I'm going to think about that one. Okay, so those are the stimuli. Role of the hypothalamus in the endocrine function, I think sorry, I'm drinking my breakfast. I think we've pretty much covered that. So the hypothalamus is going to do 2 things. It's going to influence the anterior pituitary through tropic hormones that it releases through the hypophyseal portal system of the infundibulum. And then to the adenohypophysis, and then the neurohypophysis. So the neurons that are going to come from the paraventricular and supraoptic nuclei down through the infundibulum and into the posterior pituitary, or the neurohypophysis, then the hypothalamus would release oxytocin and ADH. Okay, so that's good. All right, process is controlled by hormones. There are 5: reproduction, growth, and development, maintaining the body's defense against stressors, electrolyte, fluid and electrolyte balance, and mitosis. So those are, aren't those the 5? I think those are the 5. Nutrient, electrolyte, and fluid balance. Nutrient, electrolyte, fluid balance. Resistance against stressors, growth and development, reproduction, and mitosis. Yep, everything fits into those 5 categories. So it would be good to know which hormones go with which processes. Nervous system versus endocrine system, that's right at the very beginning of the notes. A comparison between the function of the nervous system. Well, they have the same function, homeostasis, because this whole semester has been, let's maintain homeostasis. So the nervous system will do that through the use of cellular signals, so action potentials or nerve impulses traveling down the neurons, crossing the synapse. The stimuli will be localized because the neurons are plugged right into the target tissue, and a short duration. The stimulus is only going to last as long as the neurotransmitter is there and that the nerve impulse keeps coming. But if both of those are gone, then you're not going to have a stimulus anymore. Whereas, as we already said, with the endocrine system, that the signal is chemical this time in the form of a hormone that's going to be traveling through the bloodstream. So there's no direction to that. You just have to keep circulating that hormone through the blood until it gets to the target tissues in adequate amounts to produce the stimulus that you want. I mean, the reaction that you want. And they last a long time because it has to be removed from the bloodstream. Okay, I think that's it. So does anybody have any specific questions on the endocrine system? All righty, then we'll move on to blood. All right, so what are the constituents of blood? Plasma and formed elements. The formed elements are going to be the erythrocytes or red blood cells, the leukocytes or white blood cells, and the thrombocytes or the platelets. Yeah, we'll go through their functions as we go through those characteristics. All right, so red blood cells then are biconcave discs. They are the most numerous. Hematocrit would be 45%. They carry oxygen. Because they are anucleate, they have a limited lifespan. They'll be trapped in the spleen, red blood cell graveyard, and their heme will be recycled. Well, the iron will be recycled. The heme will be eliminated in the bile by the liver, and then the globin, the amino acids, will just get made wherever we need amino acids. Capillaries are as wide as the red blood cell. I think that's all I need to know about that. White blood cells come in 5 kinds, 2 groups of those 5 kinds, granulocytes and agranulocytes. The granulocytes are the phils, so the neutrophils, eosinophils, and basophils. And the agranulocytes are the lymphocytes and monocytes. And as far as total number, we're talking 1,000 times more than 1,000. No, 1,000 times less. Yeah, 1,000 fewer per cubic millimeter of blood. So microliter of blood. Not cubic millimeter. That's a milliliter. Anyway, microliter of blood. So 1,000th of a milliliter. 4,800 to 11,000. Let's see. Their order of abundance. I don't know. What's the most abundant? Yeah, I guess the number of abundance. So never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Let's see. We will talk more about those when we get specifically into immunity. So we'll come back to them. And we'll talk more about thrombocytes as well when we get into hemostasis. So, and remember that thrombocytes are only fragments of megakaryocytes. They are not entire cells. Okay, functions of blood. 3 functions of blood. They are a transport medium for nutrients and wastes and distributing things, and they have a protective function, and they maintain homeostasis, fluid levels, nutrient levels. So those are the functions of blood. All right, red blood cell production occurs in the red bone marrow through the stimulus of erythropoietin that's made by the epithelial cells of the kidney when the kidney is hypoxic. You need heme, and so you need iron, and then you'll get a hemoglobin molecule that will have 4 hemes and 4 globins. And they're made every 120 days. And I think that's it for that. Plasma proteins, there's a whole ton of plasma proteins, but the most abundant plasma, and most of the plasma proteins are made by the liver. Of course, with the exception of the antibodies, which are going to be made by the B lymphocytes, or plasma cells that came from a B lymphocyte. Let's see. What other plasma protein? So the most abundant plasma protein is albumin, and albumin's function is to maintain colloid osmotic pressure of the blood. So I'm still drinking. So when hydrostatic pressure of the blood forces fluid out through the capillaries, then we know that it will be brought back by colloid osmotic pressure in the presence of albumin, since it is too big to be filtered through the capillary wall. Other plasma proteins would include the clotting proteins, which are all, well, most are made by the liver, and LDLs and HDLs, shuttles, those kinds of things. Okay, hemostasis. So hemostasis is the blood's ability to plug up a hole in the blood vessel so that the vessel can repair itself and blood will not leak out. There are 3 stages to hemostasis. The first stage of hemostasis is vascular spasm. So when there is damage to the smooth muscle, vascular smooth muscle, then it will contract, reducing the diameter, reducing the lumen of the vessel, and thereby allowing less blood to leave. Pain receptors will also, in the event that the vascular smooth muscle isn't damaged itself, then pain receptors can also stimulate that vascular spasm to take place. Okay, then the second step is a platelet plug. So this is where the platelets are going to cling to the breach in the wall of the capillary. Now remember, capillary walls are very, very smooth, that they're lined with simple squamous epithelium. And remember that a capillary is only simple squamous epithelium. So the reason why we don't get clots happening abundantly where we don't want them, or just spontaneously when we don't need them, is because of that smooth endothelium. But when you damage the endothelium, that exposes the collagen fibers that are in the basement membrane, and that's what the platelets will stick to. And once the platelets stick, then they activate like platelet growth factors and platelet derived growth factors and other kinds of things that will help with the clotting process and the healing process. But mostly we need the platelets to stick to each other. And then as the platelets stick to each other, we have that temporary platelet plug that allows the clotting factors to become activated and go through the rest of the cascade of coagulation. The platelets also contain actin, so we can get retraction of the clot once we start repairing that, so that we can pull the sides of the damaged area together and make it easier for the tissue to replace itself. Let's see, what else? I think that's all for the platelets. And then the third step of hemostasis is coagulation. And in coagulation, you have 2 pathways to start you have 2 pathways to start coagulation. The first step of coagulation is the formation of prothrombin activator. So the intrinsic pathway is the longer pathway, which doesn't have trauma to the blood vessel, so that the blood is leaking out. There hasn't been surrounding tissue injury. We just have some damage to the endothelium, and we start that clotting process so that we can repair that. And it takes much longer to get to prothrombin activator from the intrinsic pathway. But if you have broken open a blood vessel and we go through the extrinsic pathway, then the damaged tissue will release tissue factor, which will then bypass a lot of the cascade to get you faster to the formation of prothrombin activator. So remember that floating around in your bloodstream, you have two plasma proteins, two clotting proteins. They are prothrombin and fibrinogen. And so prothrombin needs to be turned into thrombin. And so we just made the activator for that. We just made prothrombin activator in step 1 or phase 1, so that then prothrombin will be converted to thrombin. And then thrombin is necessary to convert fibrinogen to fibrin. So fibrin right now in your body, as long as you don't have any need to clot your blood, is soluble in the plasma. And so first of all, we need it to be insoluble. So in the presence of thrombin, fibrinogen becomes fibrin, which is insoluble. And so now little stringy precipitate starts to come out. And it's not just that we need the precipitate to come out, now we need it to stick together. So then the presence of factor 13 and calcium, then the fibrin molecules will actually link together and make a mesh that will then trap all the stuff in blood and keep the blood from leaking out into the surrounding tissue. Okay. Did I leave anything out in that? I don't think so. I think that was everything. Okay, blood types. So there are 8 blood types based on the presence or absence of antigens on the surface of the red blood cell. In the first blood typing group, in the ABO group, we have, in the Landsteiner ABO group, we have type A, so A antigens present on the cell, B antigens present on a cell, AB antigens present on a cell, or no antigens on the cell to give you type O blood. If you are type A, then you have A antigens, but you will make B antibodies, so you will fight off type B blood. If you are type B, you will make A antigens. If you are type AB, you will make no antigens. And if you are type O, you will make all of the both of the antigens. You'll make type A and type B, or A and B antigens. Okay, then there's the Rh factor. So the Rh factor only comes in one version, and it's either present or it's not. So if you are Rh positive, then you do have the Rh factor antigen. If you are Rh negative, then you don't. And we need to be careful with Rh negative moms that after their first pregnancy, when the placenta is coming away from the mother, that there's an opportunity for, if an Rh negative mom has an Rh positive baby, if that baby's blood gets into her, then she's going to form antibodies in memory against the Rh positive of the baby. And so the first baby's fine, nothing's going to happen to that. But subsequent babies, because the mom now has made antibodies, IgG antibodies, which can cross the placenta, they'll cross the placenta and attack the baby's blood and give it erythroblastosis fetalis. Is that how? Yeah, I think that's what it is. I think that's how it's pronounced, the word. I know that's how it's pronounced, but yeah, erythroblastosis fetalis, where it will actually attack the fetal blood. Okay. That's it for blood. Does anybody have any questions on blood? Okay, blood flow through the heart. So please know how blood would pass through the heart. So It would enter the heart either through the superior or inferior vena cava or the coronary sinus into the right atrium, through the tricuspid valve, through the right ventricle, through the pulmonary submanular valve, the pulmonary trunk, to the right and left pulmonary arteries, to the lungs. It'll come back through the pulmonary veins, into the left atrium, through the bicuspid valve, into the left ventricle, up through the aortic submanular valve, and out the aorta. So just remember all of those parts. Let's see. Probably, I can't remember if there's a question about this. Remember that tricuspid valve or semilunar valves, tricuspid, bicuspid, or mitral valve have chordae tendineae and papillary muscles associated with them so that the valves don't prolapse during ventricular contraction, and semilunar valves do not have that. Intrinsic conduction system. So these are the non-contractile cardiac muscle cells within the myocardium. These cells, of course, are, I should have put that first, are striated, single nucleate, cylindrical with branching cells, definitely branching cells because they have to connect to others in order to propagate that signal. There'll be gap junctions at the intercalated discs, and that will allow the flow of sodium across from one cell to another. So there is a cluster of cells in the superior area, superior portion of the right atrium called the SA node that has the ability to independently depolarize due to the hyperpolarized sodium channels that once the they're going to open at a potential much lower than threshold, so they easily open and slowly open and allow a slow passage of sodium to enter the cell. And then depending on what is the intrinsic rate of contraction of that particular the SA node or the AV node or the Purkinje's, and as it depolarizes, then the cells will slowly get to threshold, which will then open the calcium channels, and the calcium will rush in and cause, well, then the rising phase of the action potential and the intrinsic conduction system cells, and then the calcium or the potassium channels will open up, and then it'll repolarize, and then we do it all over again. And if we're in the SA node, we're going to do that 60 to 100 times a minute. If we're in the AV node, we're going to do it 40 to 60 times a minute. In the Purkinje's, then we'll only do it less than 40 times a minute. So the Purkinje's will not depolarize rapidly enough to keep you alive. And then they'll also probably send you into fibrillation too. Okay, a heart block is going to come from, well, let's go back a step. So the SA node is going to send the signal to the AV node, and the AV node is going to hang on to the signal for 1/10 of a second because there are fewer out cells than there are in cells in the AV node. And because SA node doesn't just trigger the AV node, but it also stimulates contraction of the atria. So we've got to get the atrial contraction complete before we start ventricular contraction so that all the blood is now down into the ventricles. So the AV node will hang on to that, and then it'll pass it on to the bundle, or the AV bundle, to the bundle branches, and to the Purkinje fibers. Now, if for some reason we've had ischemia in the AV node and now the cells are not able to connect with one another because the gap junctions have closed and the cells are not able to receive the signal anymore, you're going to not be able to propagate that signal down to the ventricles. So you're going to get a heart block. And you can have various degrees of heart block based on how severe the damage is at the AV node. And then we're going to have to put a pacemaker in order to keep it contracting like it's supposed to, because we need the ventricles to contract, and it's really the AV node that tells the ventricles to contract, because it receives a signal from the SA node. So the SA node, if it goes into fibrillation, we don't even defibrillate for atrial fibrillation. We just try to stop it with medication. So it's the AV node is what's the most important as far as, you know, getting the ventricles to contract. And if it can't do it on its own, then we have to pace it. Okay, microscopic cardiac muscle tissue. I think I've pretty much been through that now. So the cells are cylindrical, striated, branching fibers, single nucleus, intercalated discs, so that the flow of sodium can go from cell to cell, so that all the cells that are being stimulated at a time can contract. So we get the ventricles contracting at the same time. We get the atria contracting at the same time, and not just anybody doing whatever they want, because then that's fibrillation, and fibrillation does not pump blood. The heart is contracting, it just is not contracting in a coordinated manner so that you can actually pump blood. So fibrillation is bad. All right. Another thing that's bad is tetanus in the heart. So we cannot allow the stimuli to completely contract the heart and make it stay contracted, like in wave summation and tetanus. So we have an absolutely refractory period that's much longer than what we have in skeletal muscle, so that we do not get tetanus. And the structures that allow that to happen are the slow calcium channels that we find in now the muscle tissue of the heart. So we have regular sodium channels that are going to become very permeable to sodium, and then very quickly be impermeable to sodium, so it shoots up, the permeability graph shoots up and comes right back down again. And then that depolarization is going to cause the calcium channels to depolarize, and that depolarization, the calcium channels are slow to close, so we have calcium entering and also stimulating the release of calcium from the SR in the cardiac muscle cells. And so the cell will stay depolarized for a much longer time before the potassium channels will open up and then repolarize the cell. So that way we don't have to worry about tetanus happening. Okay, so that's absolute refractory period. And so we don't even have a relative refractory period in the heart. It's just absolute refractory period, contract, absolute refractory period, contract. All right, cardiac output. So cardiac output, of course, is heart rate times stroke volume. So you need to look at those aspects that are going to regulate heart rate, and then those aspects that are going to regulate stroke volume. So, and then also cardiac output is the amount of blood that travels through a given spot in a minute. And it just so happens to be, under normal resting conditions, your entire blood supply. So you circulate your blood how many times? So how many times are there in a minute? You know, if your heart rate's 60 times a minute, then whatever. Anyway, however many seconds, I guess there are, well, no, in a minute. So yeah, how many times have you circulated? However many minutes there are in a day. Let's see. Okay, so let's do, because the next bullet says EDV, ESV, and stroke volume. So we'll do control of stroke volume first, and then we'll do control of heart rate. So stroke volume is the difference of the end diastolic volume, which is the amount of blood that enters the heart, and systolic volume, which is the amount of blood that remains in the heart after ventricular ejection. Typically, the end diastolic volume is 120 milliliters will enter the ventricles, and then, or the left ventricle, and then 50 milliliters will be the and systolic volume. So that's how much will stay. So the amount that leaves, the stroke volume will be 70 milliliters. But we need that to increase if we're going to increase the amount of or increase our cardiac output. So we don't just want our heart rate to go up, we want the amount of blood that's going to be pumped per beat to go up as well. So how are we going to do that? We're going to do that a few ways. First of all, we need to remember the Frank-Starling law of the heart that says if you increase the degree of stretch within the heart, so if we are stretching the sarcomeres, we can optimize the amount of myosin and actin that is combining and pulling. And so increasing the stretch of the heart, you will get greater force. So how do we get greater stretch of the heart? Is by increasing venous return. So let's put more blood back into the heart. So how do we get more blood back to the heart per cardiac cycle, per beat? And that's by increasing our venous return through increased skeletal muscle contraction and through the respiratory pump. Because at any one time, 65% of our blood is going to be in the venous system. And so by contracting the skeletal muscles and squeezing the blood past the venous valves, that will get more of the blood back to the heart, as well as once it gets into the thoracic cavity, into the superior and inferior vena cava, then the pressure changes within the thoracic cavity during breathing will milk the blood back to the heart as well. Since there is virtually no blood pressure in the vena cava themselves, they're just like tubes, with flimsy tubes with some water in it. Okay, so by increasing EDV, increasing venous return, then we activate the stretch of the heart, the Frank-Starling law of the heart, and we get greater contraction. And greater contraction is not only did we have more blood come in, but greater contraction is also going to decrease end systolic volume. Oh, and then longer fill time. I forgot about that one. If you can pump more blood or move more blood per contraction with your venous return, then you don't need to beat as often, and you will have a greater fill time, and that will also increase EDV. So increased venous return, increased fill time is how do we increase EDV? And then how do we increase ESV? Is by increasing the stretch of the heart, so increasing contractility that way. Preload, there we go. I knew there was a word for that. So either through preload or through sympathetic stimulation. Because sympathetic stimulation makes, epinephrine makes the cardiac muscle cells more permeable to calcium, and therefore they will contract harder. So by contracting harder, more blood is ejected from the ventricles, and then our ESV will go down. All right, so that's how can we change stroke volume. But what about heart rate? So in our brain, in our medulla oblongata, we have cardiac centers and vasomotor centers. So we have two cardiac centers. We have a cardioacceleratory center and a cardioinhibitory center. The cardiac-acceleratory center is connected to sympathetic nerves that will go out and increase permeability, well, by secreting norepinephrine on the SA node. Then it makes the cells more permeable to sodium, and they will beat faster. So they'll have a chronotropic effect. They'll make a beat more quickly. And then yeah, that's all. I was just trying to look at that picture of the cardioacceleratory center. Okay, so that's cardioacceleratory center, sympathetic stimulation. Cardioinhibitory center is parasympathetic stimulation. So when we want the heart to slow down, then we'll increase the amount of vagal stimulus on the SA node, which makes the SA node with its acetylcholine more permeable to potassium. And so the potassium will hyperpolarize the heart and therefore slow it down. Okay, then what else? I guess that's all. Then we'll talk about the vasomotor center when we get to blood pressure. Okay, heart sounds are from the closing of the valves. So the lub sound of the heartbeat is from the closing of the AV valves, and the dub sound is the closing of the semilunar valves, or the aortic and pulmonary valves. Okay. All right, ECGs. ECGs are recordings of the electrical activity of the heart. We have 4 deflections from the isoelectric line, which are the P wave, the QRS complex, and the T wave. The P wave indicates ventricular, nope. The P wave indicates atrial depolarization. So we know that just prior to the P wave, the SA node will have just fired to get the P wave, or to get the atria to depolarize. Then we know that the signal will pass down to the base of the heart, where we're going to get down through the interventricular septum down to the, to the apex of the heart, to get the Q, R, and S complex for ventricular depolarization. And then we have ventricular repolarization with the T wave, but we do not see atrial repolarization on a graph, on an ECG. Okay, then there's lots of things we can tell from an ECG, but I don't, we can tell the rate of the heartbeat on an ECG, and then we can also detect any abnormalities in the timing of the depolarizations and repolarizations. Okay, what else? Let's see. ECG, ischemia. Ischemia is the death sentence to the heart. If you do not have the proper oxygen supply to the cardiac muscle tissue, then it will not be able to produce the ATP that is so needed by the heart. Can't pump the calcium back into the SR and back out of the cell, and gap junctions close, and tissue becomes electrically isolated and no longer will participate in that contraction, and therefore weaken the contraction that the heart is able to produce. Okay, any questions on the heart? All righty. Okay, let's go on then. Blood vessel types and structure. So in the circulatory system, then the circulation part, we have arteries that are going to carry blood away from the heart. They're going to start out as elastic arteries, which are the largest ones that are closest to the heart, that have more elastic fibers in their wall so that they can accommodate that extra pressure from the ejection of blood through the heart. So we need the aorta to be very elastic and not really, we don't want it to become constricted. We don't really want it to respond to vasoconstriction, because the heart is generating that pressure, and it's still close enough that we're not going to have too much resistance so that we eliminate flow. And then we will go into the distributing or muscular arteries that do have a more muscular wall to them, so that pressure can be maintained, so that we maintain mean arterial pressure that is getting the blood out to the tissues. And then we have arterioles, and arterioles are almost always vasoconstricted to some degree to propel the blood, or to create adequate force to get the blood to the capillary bed. So we have elastic arteries, muscular arteries, arterioles, and then capillaries. And then capillaries will come, so we've been branching after the aorta down to the capillary level, and now the capillaries will combine to be the tributaries of veins. So we'll just come back together and come back together. So we become venules, and then we'll be small veins and large veins. And the large veins, well, the small veins too, but the large, primarily the large veins is where we're going to see that 65% of the blood. So the capacitance vessels is what they're called, because they're so big around in there and they're so flimsy that the blood just kind of pools around in them. Okay, well, arteries and veins have 3 layers to them. They have oh my gosh, tunica externa. So that's the outermost layer, some epithelium and connective tissue outside of there. Then we have the tunica media, which will be the smooth muscle layer. And remember that the tunica media is innervated by sympathetic stimulation, and it is much thicker in an artery to maintain pressure as it moves out towards the tissues than we find within the veins. And then we have the tunica intima or tunica interna, which is the simple squamous epithelium and its basement membrane. And then remember, of course, that capillaries are only simple squamous epithelium and they're in the basement membrane of those cells. Capillaries come in 3 types: continuous capillaries, where we don't have any breaches through the wall, that we don't have any fenestrations or sinuses or anything like that. So they are the most impermeable. I don't know. We still need exchange to happen, but they're the least leaky, I guess, if you want to say that. So we're going to find our muscles are going to have continuous capillaries. Our brain is going to have continuous capillaries. Skin will have continuous capillaries. But then the second type, the fenestrated capillaries, those are ones that have pores in them, and so they are going to be adapted for or structured for filtration and absorption. So we'll find fenestrated capillaries in the villi of the small intestine, and also in the glomeruli, the most important one. So in the glomeruli of the nephron, we will also have fenestrated capillaries. And then the last type is the sinusoid kind, and those ones have big spaces where we have to move lots of material or big material, like red blood cells that never could get through the blood vessel wall. And those will be not in your brain. They'll be in the spleen, they'll be in the liver, they'll be in the bone marrow, where we are processing that blood and need stuff to flow through sluggishly, and then have a lot of movement of material back and forth. Okay, so that's 48 minutes, which is, you know, pretty good to stop at blood pressure regulation. So, so if you, and because we haven't been over this stuff for a really, really long time, hopefully that helped to kind of focus your learning, but see how I just went through the basics of it. I didn't get real specific, but I just went through the main concepts that we have in each section. And so that's what you should be doing too. If you can think of everything that there is, in your brain about each one of these topics, then it's pretty much, the general stuff that will be on the exam. Ella just asked, are we going to meet tomorrow? No, we are not going to meet tomorrow. We'll meet Wednesday and we'll Kahoot on Wednesday, but we're not gonna, no, there is no Kahoot for reproduction because it's the easiest test. So there is no Kahoot at all. I haven't ever even made one before in my life. So we won't be Kahooting for that, for the test that's due today. Okay. So come on Wednesday, we'll Kahoot and see how we do. If there's anything as you're going through this list of stuff, anything that you're like, well, I don't know, I don't remember this, or whatever, we learned it during COVID-19, I don't know. But anyway, so there you go. Just work on, after you get today's test finished, then work on reviewing these topics. And so Thursday will be the exam. It'll be at a set time, but I'll put all that information in the announcements, because I've been talking to some other people that are like, no, I haven't been watching the videos. I'm like, why am I even coming then? It's because you guys are here. So the test is Thursday, the final is Thursday, and it will only be on Thursday. And if you miss it on Thursday, then you're so sad. Too bad, so sad, because you won't get it open anymore. So you've got to take it when I have it open. Okay, so we'll see you, I guess, on Wednesday. So have a good day tomorrow. You can go do whatever, study for some more things, and we'll talk to you on, let me just get out of here and we'll stop sharing this. Okay, so we will talk to you on Wednesday at 9 o'clock. And I'll just wait and see if there's any other questions before we go. I have not set that up yet, Kaden, but that'll be in the announcements. So once I finish thinking everything through, then you will have the time for the final. And you guys have got to accommodate me. It's a final exam, and we can't just say, oh, I can't do it then. I can't do it then. No, you have to do it then, because it's a final exam. And you will be given that time to do it, and then that's it. Just like if you were here on campus, you would just be taking it at that time, and that's it. So, so free up your lives. And not that I'm getting after you, Kaden, I'm just telling everybody that just make yourself available on Thursday. Okay, so I guess that's it. So we'll see you on Wednesday. Have a great day. Bye.